Hi, this is Dr. Mead. This is regulation of cell division. This is Kim Foglia's PowerPoint. So we're going to talk about how cancer occurs, how that's related to the cell cycle, and how the cell cycle controls cell division. So in order for an organism to function, cell division in all the systems of the body has to be coordinated. And if you think about it, how else would we grow evenly? Okay, your arms couldn't grow faster than your legs, and your brain can't grow faster than your body's ability to manage new tasks. So everything needs to be coordinated. And that's critical for normal growth and maintenance. So all the cell division timing is coordinated, as well as the rates of cell division, how fast it happens. So not all cells can have the same cell cycle. And we talked about this in one of the other videos, that your fingernails, hair, skin cells are going to go through the cell cycle much more rapidly than liver cells. So as we're saying, the frequency of cell division varies by the cell type. An embryo is going to have a cell cycle less than 20 minutes. So going from a single cell to a baby is going to require a pretty rapid cell cycle. Skin cells are also considered pretty fast. They are on a 12 to 24 hour cycle. Liver cells can still divide, but keep it in reserve. And we talked about how liver cells respond to injury. But other than that, they might not necessarily divide, but they're going to divide every year or two. That's very different than the skin cells. And nerve cells and muscle cells don't divide after maturity. They're permanently in the G0 phase. All right, so let's talk about how this is controlled. All right, there's two irreversible points. What does irreversible mean? There's no turning back. One is the replication of genetic material, okay, going from the single-stranded to the double-stranded chromosome, and the separation of the sister chromatids. Okay, so those are the two irreversible points. Okay, and then we're also going to talk about checkpoints. Okay, how does the cell assess itself, all right, and stop the process if necessary? And what happens if the process should be stopped and it's not? All right, so we're going to do a little checkpoint there, a checkpoint there. All right, so the checkpoints are what we're going to talk about. The cell cycle is controlled by stop and go chemical signals at critical points. The signals indicate if the key cellular processes have been completed correctly. Right? It's a chemical checkpoint system. All right, we'll talk about these checkpoints now. So the three major ones that you have to know are the G1/S checkpoint, which is right here, the start or restriction point. Can DNA synthesis begin? All right, this checkpoint is considered the most important checkpoint in mammals. So if you get the go-ahead here, you're pretty much going to go through the rest of the cycle if you're a mammalian cell. And if you get a no at this checkpoint, you're going to go into the G0 phase and hang out there. The next one is the G2/M checkpoint, okay, which is right here between the second gap phase and mitosis. And this is a checkpoint to determine if the DNA synthesis has been completed correctly. Okay, obviously, if the DNA is not correct, it's not a good time to make new cells. Okay, if you get the go-ahead, you're going to commit to going through mitosis. And thirdly is the spindle checkpoint, which is right here between mitosis and cytokinesis. Okay. Are all the chromosomes attached to a spindle so that the sister chromatids can separate correctly? All right, so let's talk about the G1S checkpoint. As I said, this one is the most critical in mammals especially. It's the primary decision point. It's called the restriction point. If the cell receives the go signal, it divides. And this is the internal signals for cell growth, for cell nutrition. And the external signals okay, we're going to get into the internal and external signals along the checkpoints in a couple minutes. Okay, if the cell does not receive a signal, it's going to exit the cycle and switch to the G0 phase. And a lot of cells are hanging out in the G0 phase. All right, so that boing sound is for the red light. Okay, and that's the non-dividing working state of the cell. So to say a little bit more about the G0 phase, okay, or as it's called, the resting phase. It's a non-dividing differentiated state. Most of our cells are in the G0 phase, like the liver cells, which are in G0 but can be called back to the cell cycle by external cues. We talked about how the liver responds to there being damaged cells there. And then nerve and muscle cells that are highly specialized and arrested in G0 and can never divide again. Activating cell division. How do cells know when to divide? So the reason that we're talking about mitosis along with the membrane and with cell signaling is because of the signals that are involved here. Right, cell communication signals. Chemical cells in the cytoplasm give the cue. And when we are saying signals, we usually mean proteins. All right, we're talking about activators and inhibitors. So this experiment here is on page 228 in the book. And the question that this experiment is answering is, are there molecular signals in the cytoplasm that regulate the cell cycle? So they fused a cell that was in mitosis, the M phase, with a cell in G1, and when fused, the cells both become in M phase. And they also took a cell in S phase and fused it with a G1, and both cells entered the S phase. So the conclusion was that there must be molecules present in the cytoplasm of cells in the S or M phase that control the progression of the phases. So once those signals are there, they're going to move those cells along. So we're talking about the go signals, do it signals, the protein signals that promote the cell growth and division. And we have internal signals, which are the promoting factors, and we have external signals, which are growth factors. And this is the primary mechanism of control. We have phosphorylation, which we talked about in I forget the name of the chapter, but the cell signaling chapter, and the kinase enzymes. We're talking about either activating or inactivating the cell signals. All right, so this is from page 229 and 230 in the textbook, the cyclin-dependent kinase and cyclin, which are the cell cycle controls. So cyclins are regulatory proteins. So protein kinases are the enzymes that activate or inactivate other proteins by phosphorylating them. The kinases that we're talking about here that drive the cell cycle are present at a constant concentration in a growing cell, but often in the inactive form. To be active, the kinase has to be attached to the cyclin, a protein that gets its name from the cycling fluctuation in the cell. So that makes these kinases are special, and they're called cyclin dependent, or CDKs. So the activity of the CDK will rise and fall with the changes in the concentration of its cyclin partner. So if this part's a little bit confusing, we're talking about page 229. So this is just like the other kinases we've talked about in the past, but this one is special, the cyclin dependent, because it's dependent upon being attached to this cyclin, and the cyclin is going to have fluctuating concentration in the cell. So that's how it levels the cycle in the cell. The CDKs, the cyclin-dependent kinases, phosphorylates cellular proteins. So it's activating or inactivating proteins. And then together, the CDK cyclin complex, okay, here is the phosphates, the phosphorylation to activate it. Okay, so we have an inactivated and activated CDK, so you can see the difference there. So this triggers the passage through the different stages of the cell cycle. Okay, this picture comes from page 230 in your textbook. These are the researchers involved, and this is showing you the interaction of CDKs and the other cyclins. The other cyclins triggers the stages in the cell cycle. So the point of this is to show you that there's a relationship between the MPF, which is the maturation promoting factor, MPF, but your book suggests thinking of it as the M phase promoting factor, so something that's going to promote mitosis. And so they're showing you the relationship between the concentration of MPF and cyclin. So MPF, the M phase promoting factor, triggers it past the G2 into mitosis. So when the cyclins that accumulate during the G2 associate with the CDK, the result, the MPF complex, initiates mitosis. Phew, okay. So there's a lot of different kinds of cyclins, and they each have a specific role, and cyclins are pretty unstable. Oscillations in the activity of cyclin-dependent kinases, the CDKs, dictate how the cell progresses through division. So for most organisms, the cell cycle machinery relies on the CDKs. And the coordination of the CDKs are responsible for a lot of the cell cycle events. So this is a diagram to show you the molecular control of the cell cycle. So I think this is going to fill in as we go here. Okay, so there's the G1/S checkpoint. Okay, during the G1 is the growth factors, nutritional state of the cell, and the size of the cell. So moving from inactive to active. All right, I'm going to fill in all these blanks here so we can put it all together. So this goes along with the cell cycle chart on page 230 of your textbooks. So this is to put together the molecular processes and signals between inactive and active along the way. So these are the three checkpoints that we talked about, the G1S, the G2M, and the spindle checkpoint, and then the molecules that are involved. So MPF, the mitosis promoting factor, is the one that we just talked about. And we're going to talk a little bit more about the anaphase promoting complex in a second. Okay, it won't move forward. Come on. Come on. Hang on. Something wrong. Oh, there we go. All right. Oh, they were just adding cytokinesis. I don't know why that wouldn't advance. There we go. All right. The cyclin and cyclin-dependent kinases. Okay, so CDK and cyclin together form an enzyme that activates other proteins by phosphorylation. The amount of CDK molecules is constant during the cell cycle, but their activities vary because of the regulatory function of the cyclins. So CDK can be like an engine, and cyclin is like a gearbox controlling whether the engine will run in the idling state or drive the cell forward. So just to give you that again, CDK is like an engine and cyclin is like a gearbox. So the cyclin is going to decide whether or not the engine's just going to hang out and idle or move forward. And what they're telling you here is the proper regulation of the cell cycle is so key to life that the genes for these regulatory proteins have been highly conserved through evolution. The genes that we're talking about for regulation of the cell cycle are the same in yeast, insects, plants, and animals, including us. So this is a pretty strong standing part of evolution in a lot of different organisms. And so we're talking about the relationship between cyclin and CDK. All right, so moving on to the external factors that affect the cell cycle. So growth factors are proteins that are released by certain cells that stimulate other cells to divide. So some cells are giving a chemical message to other cells to begin dividing. And the picture being shown here is like the picture on page 232, showing you that that some cells have a limit to how many can fill up a particular space. And this is called density-dependent inhibition. So what they did was lay out a layer of cells until it grew and filled a single layer, and it stopped dividing right here. And then they scraped some away so that there'd be a little bit of hole, and then they filled in again just to the single layer. And this is in normal mammalian cells, and the reason we're saying normal is because this is not what cancer cells do. So the availability of nutrients and growth factors and a place for attachment limits how dense a bunch of cells can be. So that is part of the external factor. So how much space there is, how much nutrients there are. That's called density-dependent inhibition. So inhibition meaning that the growth is inhibited because they can only be so densely packed. So if you've got overcrowding, they're not going to divide anymore. So each cell binds a bit of growth factor. And there's not enough activator left to trigger division in any one cell. Right, and then we have the other part of this is anchorage dependence, because the cells have to be attached to something. And they have receptors to figure out where they can be and how much further they can grow. So the reason, again, that this is included in our whole signaling part is that look at how growth factors work. All right, so here's a receptor and here's the growth factor. It's a signal, of course, in the membrane. So this is a protein. So we don't have to know every detail of this, but when the growth factor signals connect with the receptor, it's going to cause a signal to end up going into the nucleus to tell the cell to divide. An example in your book has this experiment as well, the platelet-derived growth factor. Right, so it's a molecule made by the platelets in blood clots, and the binding of platelet-derived growth factor to cell receptors stimulates the cell division in connective tissue. So this experiment's on page 231, and they took a sample of connective tissue and cut it up into small pieces, and then they used enzymes to get rid of the extracellular matrix so that you just ended up with these free-floating cells. And then they transformed them into two trays, and one they put platelet-derived growth factor in and one without, and the one without didn't divide and the one with went on to divide, proving that this platelet-derived growth factor is a growth factor and external signal leading to cell division. Okay, and this is how wounds are healed. Okay. Don't forget to mention erythropoietin, EPO. Not sure why. Let me think about it. Erythropoietin is a hormone made by the kidney that promotes the red blood cells to form in the bone marrow. It's a glycoprotein. Kidney cells that make the erythropoietin are specialized and sensitive to low oxygen levels in the blood. The cells release the EPO, the erythropoietin, when the oxygen level is low in the kidney. The erythropoietin then stimulates bone marrow to make more red blood cells and increases how much oxygen can be carried in the blood. So the erythropoietin is the main regulator of red blood cell production. So it's an external signal that's telling cells to divide. So the major function of erythropoietin is promoting the development of the red blood cells and making more hemoglobin to transport more oxygen. So we're going to talk a little bit about cancer, and then we're going to be done talking about the cell cycle and mitosis. So what causes cancer? Well, growth factors can create cancers. All right, call them, there are these proto-oncogenes. Proto-oncogenes. And they normally activate cell division. It's a growth factor gene. It becomes oncogenes. This onco root means cancer. So it becomes cancer causing when it's mutated. If it's switched to on, it causes cancer. An example is RAS, which activates cyclins, encouraging mitosis, encouraging cell division. Okay, and we have tumor suppressor genes, which normally inhibit cell division, and these are really important proteins made by the tumor suppressor genes, and if they're switched off, they can cause cancer. So these are super important genes that should be turned on to inhibit cell division of abnormal cells. And an example is P53, which is a protein that's made, and that protein is super important for inhibiting cell division. So it stops abnormal cells from growing. So these are the two examples of growth factors that can create cancers. So what is cancer? Cancer is essentially a failure of cell division control. It's unrestrained, uncontrolled cell growth. So remember how we talked about the density-dependent inhibition where we had a single layer of cells? Well, when that doesn't occur, cancer cells can just keep growing. They don't care about forming a single layer. All right, so what kind of cell division control are we talking about? All right, we're losing the checkpoint stops. Okay, gene P53 plays a role in the G1S restriction point. Okay, that one, it detects damaged DNA and can halt the cell division if the DNA is something wrong with it. Right, so the options are to stimulate repair enzymes to fix the DNA and to force the cell into the G0 resting phase, keep the cell in G1 arrest, or cause a damaged cell to die. All cancers have to shut down P53 activity. So if P53 is functioning correctly, we're going to be able to control the cell division of these abnormal cells. So for it to become cancer, it's going to have to shut down the P53. So P53 is called like the guardian, the watcher. All right. Kim's PowerPoint, she calls it the cell cycle enforcer. That's a master regulator gene. All right. We're going to move past this. Let me pause. So we just have a few more things to talk about. So for cancer to really develop, it's going to have to overcome these 6 main points. You're going to have unlimited growth. So all the growth promoter genes are going to be on. We're going to ignore the checkpoints. We're going to turn off tumor suppressor genes like P53. We're going to escape apoptosis, so the cell is not going to commit suicide. We're going to have immortality and unlimited divisions. So we're going to turn on chromosome maintenance genes. We're going to promote blood vessel growth. So these cells would like a blood vessel supply. They want oxygen. They're going to overcome anchor and density dependence, turn off the touch sensor genes so that they don't have to stick to one area. They can grow completely wherever they want, and that's how they can grow quite big and take up space in a body where those cells would not. So this is the picture from page 232 when the cells are ignoring density-dependent inhibition. So what causes this initial cell to form is a mutation. It can be caused by UV radiation, chemical exposure, radiation exposure, okay, heat, right? And so it starts with a single cell and then grows, okay? And once it invades neighboring tissue, okay, and can move to other parts of the body, well, that's what we call metastasis. And the other triggers here that didn't quite play through: cigarette smoke, pollution, age, and of course, genetics. Okay, so tumors are massive abnormal cells, and these are key terms you have to know: the difference between benign and malignant. Benign tumors are abnormal cells that remain at the original site as a lump, okay, and P53 has halted the cell divisions successfully. They don't cause serious problems and can be removed, okay? So this is not cancer, this is a tumor, okay? So it's an abnormal cells that they're staying where they are, right? Where a malignant, okay, the cells leave the original site and they have a loose attachment to nearby cells. They get blood and lymph to move on, and they start more tumors and can become what we call metastasis, where it moves from the original site out to other regions of the body, and it will impair the functions through the rest of the body. Treatment for cancer that you've probably heard about is radiation to kill the rapidly dividing cells. This is really commonly used for thyroid cancer. Chemotherapy to stop the DNA replication and stop mitosis and cytokinesis and stop blood vessel growth. So if you have colon cancer, say, then you're definitely going to undergo chemotherapy to get control over it, because that's one that's easy to go to metastasis. And then there's a lot of, beyond these traditional treatments, a lot of new drugs out there too. So that's the end of the control of the cell cycle video. Please make sure you wrote down any questions you have and watch the review video. Bye.